My pleasure to introduce Simon Laughlin, who got his bachelor's degree and master's at the University of Cambridge, and then received his PhD at Australian National University, where he also did his postdoc and stayed on for a number of years as a fellow and senior fellow before moving to Cambridge, University of Cambridge in 1984, where he's been ever since. And in 2015, he became emeritus professor of neurobiology in the Department of Zoology. He's a fellow of the Royal Society, and also he has a book called Principles of Neural Design that he's co-authored with Peter Sterling, and it's an award-winning book. I think many of you in this audience apparently have already read it. So he's focused for many years on the insect visual system. I think he started that as a PhD student, where he's unraveled using this and other systems how neural circuits integrate information and how neural function is impacted by adaptation and synaptic plasticity. And something relevant to his talk today, how energy consumption of neurons impact on their connections and their performance. And he's approached these problems in large part through a combination of using electrophysiological and behavioral approaches. And so without further ado, I'd like to introduce Simon Laughlin, and his title of his talk is The Energetic Brain. Well, thank you.
Can you hear me better now? Good. Okay, so today's talk is actually going to be rather general. It's going to focus a little bit on energy consumption at my own work, but it's going to give a background and ask in the most general terms, what is it that neurons do and how do they support behavior? But we'll start off with energy, and modern work on energy in the brain really started with Seymour Kety in 1943 or so, when with his boss Schmidt, he developed a new technique for measuring the oxygen and glucose consumption of the brain, something that had not been accurately done before. This technique is definitely something that you shouldn't try at home. It involved cannulating the blood vessels going carotid and vein and artery, and taking samples from the blood going into the brain and out of the brain, and measuring the levels of glucose and oxygen in those samples, and then by using a very clever tracer technique and a nice mathematical model, looking at the washout of the tracer to get the blood flow. And if you know how much blood is flowing through and how much oxygen is being removed and how much glucose is being removed, you can get a good estimate of how much glucose and oxygen the brain is consuming. And what he found was that the human brain consumes a very significant quantity of oxygen. While you're sitting here resting, not exercising, it's actually 20% of your metabolism, even though it's only 2% of your brain's body weight. And this immediately tells you that the brain is an intensive consumer of energy. And of course, it was known to the Greeks, and even before that if you cut off the supply of oxygen or glucose to the brain by strangulation or by asphyxia, people very rapidly lose consciousness. And very often the results are fatal, and you don't or even if you don't die, you often suffer lasting injury. So the use of oxygen and glucose, the use of energy, is critical for neural function. They also did the obvious next thing. There had been a lot of work done on the energetics of muscles, and you know that when you work out and you increase the amount of work that your muscles do, they use more energy. And so they figured, well, maybe if you increase the amount of neural work done by the brain work, you'll see an increase in energy consumption. And so they got people to do things like translate from one language into another, solve differential equations, and so on. And with their technique, they could not measure a significant increase in energy consumption. So energy consumption is ongoing in the brain and is only slightly perturbed by changes in overall level of neural activity, unless you do something drastic like apply anesthesia, or as we'll see in a minute, you start to age. So this brings us on to the topic of aging and maturation. And this is data that's very similar to the data that Kety collected in the 1950s, which shows the consumption of glucose as a function of brain's consumption of glucose as a function of a person's age. And this was replotted, and this is a sort of meta-analysis by Goyle et al. And down in this direction, the scale goes in months. So this is 20 months before birth. And up here, the scale goes in years. What? Sorry, not 20 months. That must be weeks. I must have yeah, you're quite right. Gosh, quite right. We're not elephants. Okay, or whales. Yeah, so that's weeks. Don't know how I did that. Anyway, and you can see that at birth, the consumption of glucose in the brain per 100 grams of tissue, so this is intensity, this is normalizing for any changes in growth of the weight of the brain, increases steeply, and it peaks at about 7 years of age, and then it declines, first of all quite rapidly, and then throughout your life declines slowly. And this curve here, which shows the amount of glucose that's being used for aerobic metabolism. So they measure the oxygen consumption in similar experiments, and so you can work out if the oxygen is all being used for aerobic metabolism to generate ATP by essentially burning sugar, how much glucose would that oxygen consume? And you can see that the brain is actually consuming more glucose than it's using for its energy requirements. And this has now been associated with synaptic plasticity and the growth and the formation and the maturation of the brain. But this consumption here is about 50% of a child's resting consumption at that age. So the next question that came up is really, well, is this glucose being consumed by neurons, and how does it relate to the neural activity? And the person who really cracked this problem was Sokoloff. This is him at the time that he did these experiments, and this is him just shortly before he died in 2015. And he designed a very clever technique. His clever technique was to use an analog of glucose, 6-deoxyglucose, which is recognized by the transporter protein that takes glucose into cells, but is not recognized by the first enzyme in the breakdown of glucose. So this 6-deoxyglucose accumulates in neurons at a rate that's a function of their glucose uptake. And he labeled 6-deoxyglucose radioactively, and then he could make sections and do autoradiographs. And this shows the autoradiograph of a piece of monkey's visual cortex where the monkey had been injected with 6-deoxyglucose and a flashing target, fixated on a flashing target. And you have a map of space in your visual cortex, and you can see that the pattern on the target has been transferred onto the map of the visual cortex as an increase in the uptake of glucose, as shown by these dense silver grains of this from the generated in the autoradiograph from this radioactive glucose. And this and many other experiments that he did showed that increase in neural activity, increases in synaptic activity, increased glucose consumption, and that consumption is localized as far as he could resolve in these techniques to the neurons that have increased their activity. Well, of course, the brain is consuming all of this oxygen and all of this glucose, so it must have a good system for supplying oxygen and glucose through the blood. And this slide here, which I only came across recently, is a depiction of the vasculature of the human brain, showing all of the blood vessels which have been filled with an agent and then reconstructed from sections, or maybe from scans. And if you add up the total length of all these blood vessels in the brain, they come to about 800 kilometers. So there's a very large, rich vascular system there. And of course, most of the 800 kilometers is the very smallest of the blood vessels, the capillaries, which come very close and intermingle with the neurons in the cortex, or in neuropil. And this here shows a light micrograph. That's 100 microns there. And the capillaries, which are the finest vessels formed by very small numbers of cells, the capillaries are picked out and have stained green, and around investing the capillaries are another sort of cell called the pericytes, which wrap around the capillaries, and they're stained in red. And in about 2014, my colleague David Atwell, who we'll come to in a minute, showed that in using retina, that when neurons are more active and blood flow increases, this is actually mediated by the pericytes relaxing so that the blood vessels can open out more. So the pericytes are kind of strangling the blood vessel and they relax. And it's been found, starting with David Atwell's work, but there's a lot of work going on this at the moment, that's what's happening is that when neurons in the immediate vicinity are more active, they produce chemical signals, obvious ones such as release of glutamate, but a lot of second messengers as well. They're sensed by a type of glia called astrocytes, which integrate these signals and then communicate with the pericytes to get them to relax, to increase the blood flow. So there's an incredibly intricate, fine local control of blood flow. The blood cell flow is continuously adapting due to this signaling to match local demand. And that presumably increases the efficiency with which the blood is used. And interestingly, because when you actually open up the blood vessels, the oxygen consumption in the vicinity of the neurons increases, it's reducing the lifetime exposure of the brain to oxygen, which could be a good thing for combating aging. And of course, it's this mechanism here, operating at the level of pericytes and capillaries, which is primarily responsible for producing the bold images, functional magnetic resonance images, needn't tell you anything about these, that have revolutionized cognitive neuroscience over the last 2 to 3 decades. And so this is a consequence of the local control of blood flow by this very intricate system. So we can conclude, first of all, that the brain consumes significant quantities of energy, and the energy is primarily generated within neurons by aerobic respiration using mitochondria, in which glucose and oxygen combine to make water and carbon dioxide and energy, which is then trapped and delivered in the form of ATP. And that active neurons use more energy when they become active, and that can be monitored directly by Sokoloff's, or indirectly by Sokoloff's types of technique, and also by their effect of that activity on the control of local blood supply in bold signals. And I have to say that one of the big problems in the field now is that we still do not have reliable methods for estimating the rate at which neurons are consuming energy individually. You can see changes in the level of substrates and products such as ATP, decline in glucose and so on, but you want to know how much has actually been how much ATP has been generated, how much glucose has been consumed, and to do that you have to convert a concentration that you've measured, quite simply, into a flux. And that's a difficult thing to do because you need to know what the rates, what the compartment sizes are, and what the rates of entry in and out of the compartment are, and so on. So there's a lot of interesting questions still to be asked about energy consumption at the level of single neurons, exactly how much it goes up and down. Are they fed ATP by surrounding neurons and by surround? Those are all still open questions. But of course, the big question are what are these neurons using their energy for? What are the processes that are consuming energy? And how is their use of energy related to the brain's function, which is, of course, the generation of behavior? And I'm going to talk knowledgeably about this topic, and then in a more sort of speculative general way about this topic. So for those of you who are not neuroscientists, just a little reminder that inside your brain you have about 90 billion neurons, nerve cells. They form about 10 to the 14, 100 trillion synaptic connections. And these synaptic connections are made by using what in our book we call, for simplicity, wires. These are the dendrites and the axons and the axon collaterals that enable neurons to send signals to distant synapses. And it's only recently that people have sat down and actually worked out how much wiring you really have in your brain. And the answer is rather astonishing. You have 2 million kilometers of wiring in your brain, and about 1.5 million of that is in the 500 milliliters of grey matter of cerebral cortex. And in a minute you'll see why that number is so large. And the overall energy consumption of this system is something like 20 watts. And in my next lecture, I will look to try and answer the question, or give some answers to the question, why is this consumption from a very powerful computer in your brain so much smaller than the consumptions of digital computers, and in particular supercomputers? But we'll go on now in the quest to see what are neurons using this energy for, and how much of the energy how is that usage distributed among its different processes. And to do that, David Atwell and I constructed an energy budget of a piece of active neural tissue, and we chose cortical grey matter from in rodents. And the reason is that at the time we did this in 2001, there was a lot of data on both the neuroanatomy of rodents, particularly mice, and also on the physiology of the mechanisms that consume energy at synapses and transmitting signals along axons. And just to remind you, if you take a section through the brain, you see that there's white matter. These are the myelinated axons, mainly of pyramidal cells, but some of afferents, which are carrying signals over a longer range between more distant cortical areas. And these signals are being processed in the grey matter, which is this thin film of synapses and neurons over the white matter, at least in the human brain. And if you take a section through the grey matter, you see a familiar picture. This one is from Cajal, of pyramidal neurons forming many, many processes. Those are the wires, and along those wires are little knobs where there are synapses. And so from the anatomy, we could work out what the length of the wiring was and how many synapses there are. And from the physiology, we could work out how many signals were being we could work out how much energy was required to send information along the wires and how much energy was being used to transfer signals at synapses. And just to remind you what's going on, then in this tissue, there are something like 10,000 neurons, mainly 80% of them pyramidal neurons, in a cubic millimeter of tissue. And they make something like 8,000 synapses, this is for mouse, per pyramidal cell, which gives you a synaptic density of about 1 billion per cubic millimeter. And in order to connect those synapses, a single cell makes 4 centimeters of these fine axonal connections called axon collaterals. I have to say, when we wrote our paper to Nature that was rejected, one of the referees said, this paper contains a serious error. 4 centimeters is longer than the mouse. So this is an astonishing number, and it was very, very clearly checked by the person who first derived it, Valentino Breitenberg, because it says that within each cubic millimeter of cerebral grey matter, you have 3 kilometers of wiring. And now I think you can see how you managed to get 1.5 million kilometers in your grey matter. So this is a really astonishing fact that you can go off and tell your friends in amazement. And of course, these wires are very, very thin. Their average diameter is about 0.3 of a micron. So the pyramidal neurons generate action potentials. They send these spike signals along the axon collateral, their axon collaterals, and at the same time, they project them along their major myelinated axon into grey matter down here. And in this talk on energy, I'm not going to talk I'm just going to tell you one thing about the consumption of white matter, that the specific metabolic rate of white matter is about 1/5 that of grey matter. So in fact, because they're not processing signals synaptically and because they're myelinated, they're using far less energy. And when the action potentials arrive at synapses, they produce smaller electrical signals, PSPs and IPSPs, with which I'm sure you're all familiar. And just like in your mobile phones, the transmission of these electrical signals requires energy. So we're just going to simply work out how much energy is required to send signals along this 4 centimeters of axon collateral per pyramidal cell. And when the action potential arrives at the synapses, how much energy is used by the synapses, both for the release of neurotransmitter and for the activation of postsynaptic currents. And the basic calculation is that we work out how many ions flow across the membrane, how many sodium potassium ions flow across the membrane when you generate an action potential per unit membrane area of axon. We know the lengths of the axons, the diameters, so we know the total area. So we can get a minimum sodium and potassium flux, and then we can work out how many times the sodium pump in the membrane has to crank over to put the sodium back into the neuron and pump the potassium back out, because that has a stoichiometry of 3 sodiums to 1 ATP. I think that's right. And so that gives you an ATP consumption from the ion flux for the action potentials, and you can do the same thing with the synapses, and you can also factor in the costs of neurotransmitter recycling and even the recycling of synaptic vesicles. And this was something that I discovered, that you could factor these things in by going to the literature, was something that I discovered when I tried to work in a paper in which we worked out how much energy was being used by fly photoreceptors and fly synapses. And I realized that there was sufficient data in biochemical and physiological studies and biophysical studies of synapses to really get some reasonable estimates of how much energy was actually being used when a vesicle of neurotransmitter is released. How much energy to retrieve the neurotransmitter, refill the vesicle, repackage the vesicle, and so on. And then I managed to persuade David Atwell to, well, I didn't actually persuade him. This is a good example of how science works when you're not thinking about it. We both had to examine, we examined together a PhD thesis. And we were sitting around before the PhD just discussing, and David said, you know, Simon, there's a lot of rubbish being talked at the moment about how much energy is being used for transmitting signals in the brain, that people are saying a lot of energy is being used for neurotransmitter synthesis and turnover. And this was because functional imaging had just been discovered, or was just taking off. And if you claimed that you were working on a system which was using large quantities of energy which were linked to neural signaling, then you could claim that you were actually working on the basis of functional imaging. So you were more likely to get a grant. And we both thought this was absurd because you release a small number of neurotransmitter molecules and they bind to a channel, and the channel will pass through it something like 100,000 ions, which require a lot of energy to pump them back out. And I'd been doing this energy budget on flies and then even made a sort of proto-budget on cortex. And I said, well, Dave, I happen to have a whole lot of calculations here which suggest that you're right. And so he took them away, and he is famous for being a very astute, sharp person. And I fully expected him to come back and say, phone me up 6 weeks later and say, Simon, you know, you just simply don't understand the Nernst equation or something. But in fact, he phoned me up and he said, okay, this looks good, we'll do it. And so I was extremely fortunate in this case because David is, there he is was at the time that we were doing this, one of the leading experts on glutamate turnover in cortex, neurotransmitter turnover in cortex, and a very fine biophysicist. So together we worked out how much energy was being used by synapses and how much energy was being used by spikes. And the result we got, well, they weren't results. The calculations we made based on data in the literature was that there were fairly good measurements of protein synthesis, macromolecular synthesis, and so on, and baseline rates of energy consumption in the brain when the brain is totally anesthetized. And they suggested that about 35% of the energy consumed by cortical grey matter goes for what I've called maintenance. Now, I've put maintenance in inverted commas because, in fact, a lot of this energy is not to do with maintenance. It's actually to do with the processing of information and basically reprogramming and actively adjusting the brain and computing over lots of different timescales and so on. And in my second lecture, I will show you why that is. We found that about 15% of the energy was used to transmit action potentials. This overturned previous claims that action potential consumption was negligible. And we found that about 50% of the energy went into transmitting synaptic signals. And most of that was postsynaptic, because opening those channels, and only a small amount was presynaptic. And this is good news for the brain, because the synapses are involved in information processing, and in most instances, when information is transmitted by spikes, information is simply relayed from one part of the cell to another. There are exceptions, but in most cases it's simply relaying. And that's an overhead. That's just something you have to do if you want to process information. So this is quite an efficient system. But of course, getting these numbers still leaves a lot of questions unanswered. So how does the energy that neurons expend sending signals to synapses and across synapses support a brain's function? That's really the big question, which is, of course, the generation of behavior. And to understand that, or to start to understand that, one needs to know what are neurons and what do their signals do. And quite a bit of my final talk will be looking at that. At this point, I want to diverge a bit to say, well, we now take it for granted that we know a lot about neurons, but there's an awful lot we don't know. And the reason is that the brain is a tangled thicket of tiny cells. That if you take a piece of fly brain and you cut a section and you stain all the neurons in that section, in this case with a silver stain, Nick Strausfeld has done this, and you can identify the cell bodies and the processes and the axon tracts of all the neurons in this small little piece of fly brain, that's the central complex there, it looks like a tangled mess. And you might say, well, let's look at it at higher magnification. So if you go to the electron microscope and you look at electron microscopical sections, you see that it just looks like a section through a bowl of vermicelli. That there are some big dendrites here. These are pyramidal cells in hippocampus. There are some big dendrites. But all of these little round things here are wires. They're all axon collaterals, and dotted around in them are little densities, some of which are mitochondria, which are generating energy, and others are associated with synapses. And this is essentially the reason why brain research is pretty difficult. So how do you cut through the thicket? Well, the person who did most to cut through the thicket was Ramon y Cajal, the famous Spanish neuroanatomist, who is, in my opinion, one of the 10 finest neuroscientists who've ever lived. And Cajal took a technique which had been discovered by this person, developed by this person, literally developed, Camillo Golgi. And Golgi found that if you fix tissue, neural tissue, and then you soaked it in potassium dichromate for 2 days, and then you took it out and you washed away the dichromate, and you then developed it with silver nitrate, you got deposition of silver grains in single cells. Now, this technique simply wouldn't work if every single cell picked up the dichromate and developed. You'd just end up with a big black blotch. And what fortuitously Golgi observed was that single cells were selectively taking up the potassium chromate and were being developed to reveal their morphology. And Cajal took this technique to a new level because he was basically a more astute and imaginative and creative thinker and a better observer than Golgi. They both shared the Nobel Prize at the end of the 19th century. So Golgi did okay. He got his credit. But Golgi believed that the brain was a syncytium, that all of the neurons were joined together because, after all, it was a network, and how else can you make a network of cells because they hadn't actually found synapses. And Cajal showed conclusively that Golgi was incorrect. So even though Golgi discovered the technique that unlocked the door, he was wedded to an idea that prevented him from really seeing what was going on. And what Cajal found in a nutshell, or at least the little piece, he found many, many things. And the little pieces that interest me are, first of all, that neurons come in many shapes and sizes, and each is characteristic of the location of that neuron and its function. So here are cells in cerebral cortex from a human infant, showing here pyramidal neurons, and I think there are some interneurons around here as well. And they look like that. Now, as a computer scientists seem to think that neurons are just computational units, like they are in bits on silicon chips, like transistors and so on, or a simple integrated circuit. So there's a kind of standard neuron, and you draw a diagram of it. And that's a Purkinje cell, a single neuron in the cerebellum. It has a very different form, which we will see is related to its function. And he looked at lots and lots of different neural tissues and found in each case that there were these unique forms, and very often he could relate them to his function. And this is one of my favorites, because these are the optic lobes of the honeybee, which I've actually worked on. I worked a lot on this part here. And what you can see is that each one of these navels refers to a neuron with a different characteristic name. He identified close to 100 different types of neuron, and they form this wonderful regular arrays with interlocking branching patterns showing a great deal of spatial regularity. And this is compared with the hippocampus, which seems to be a little more jumbled. And Cajal said that if you compared the insect optic lobe with a part of the mammalian brain, a corresponding part of the mammalian brain, such as the visual cortex, it was like comparing a hunting case watch, which was the most exquisitely made 19th century watch with beautiful interior clockwork and a beautifully etched case and so on, with a cheap alarm clock. And this is the cheap alarm clock. Because we now know that, in fact, this is just as intricately wired as that. It's just that the connections are not so spatially regular because it's not dominated by the need to process visual input. Cajal's, probably his most important contribution, was to show that neurons form circuits that direct signals in a purposeful manner. And he illustrated this, first off, with these two neurons in the nerve cord of an earthworm. And this neuron is a sensory neuron, and it comes up and branches close to and forms synapses with a motor neuron, which goes to muscles on the other side. And what this means is that if I'm an earthworm here, and I've got a sensory neurons on this side, then if I give a sensory stimulus, the signal is transferred to a motor neuron on the other side, and the muscles on this side contract, and the earthworm flexes away from the noxious stimulus. And so Cajal pointed out that this was a purposeful circuit. It was capable of performing a useful behavioral function. But his best descriptions of circuits were probably in the mammalian retina, where he pieced together, patiently by looking at hundreds of individual neurons in whole mounts, he pieced this together and saw these regularities in connection. And he pointed out that these circuits were carrying signals along definite cellular pathways, and he put these arrows on, which, as there was no electronics in those days. The most sophisticated circuits were in home electrical supply, in telegraphy, and in control of machines and electrical engineering, was a very advanced idea. But more importantly, he suggested that the forms of the neurons subserved their function in the circuit. So, for example, here you have this bipolar cell collecting inputs from several of these long, skinny things. These are photoreceptors, and they're rods. And what this bipolar cell is doing is it's collecting signals from several rods and summing them together before sending them off to the next neuron. And that's increasing the sensitivity of your visual system at low light levels by summing together signals at the sacrifice of spatial resolution across here. And here he depicts what happens at high light levels. You can get the highest spatial resolution possible, which is set by the fineness of the spacing between your cone photoreceptors, showing here. And in this region, Cajal showed that there were so-called midget bipolar cells. A single cone connects to a single midget bipolar cell, which actually connects to a single midget ganglion cell to send their signals to the brain, so that you can retain the highest possible spatial acuity of your cone mosaic. So this is a neuron's form following its function and purpose. And finally, he observed that neurons connected at funny sort of knobby structures that he called synapses. And at the same time, the physiologist Sherrington, working in Britain, showed that there were physiological entities that you could infer in the brain which had the characteristics you might expect of a synapse. And it's remarkable, it's a testament to Cajal's ability as an observer and as a draftsman, that if you look at recent micrographs made using the latest molecular techniques where you can get single neurons to express a fluorescent protein, and here are some pyramidal cells and associated neurons in cortex, and that is Cajal's drawing of similar structures. And with the molecular techniques, you can really go to town and show that there are a superabundance of neurons of different forms. This is neurons in the brain of the fly Drosophila, and all stained in different colors. And I just want you to show that they all come in different shapes and sizes. And you can also use developmental and molecular techniques to show neurons forming making connections to form circuits, shown here in the nerve cord of the larval fly. And the molecular techniques can show you where the synapses are on neurons, and that identifies typical chemical synapses which release neurotransmitter to activate postsynaptic receptors. And I hope you all know about that. So the question, Cajal achieved some important got some important answers to the question, what are neurons? They're cells, they're unique to the nervous system, they're specialized to make connections. They transmit signals, and they process information by passing signals through circuits, and that they have forms that relate to function within those circuits. So that leads to the question now, how do neurons support behavior? Well, first question to ask is, and that is a $64,000 question in neuroscience. That is a huge problem that people are addressing at very many levels. And what I'm going to give you is a simple take on this that you could go and tell your granny. And she might actually have, or your grandfather, he might actually have a better idea about what the sorts of things that are going on in the brain that are being supported by neurons. But of course, just like us, most of these things we can't make head or tail of what they're doing. So my argument is that behavior is a set of connected events. We link things together, and it depends on establishing relationships between these events, so that if this thing happens, that thing has to happen, or if these two things happen together, that this relationship tells us that there's something out there that looks like this, or whatever. And I'll just give you a few examples of this basic, rather flaky-looking overview. So here are worms, Caenorhabditis elegans. They happen to be in space in this picture, and they're wriggling. And in order to wriggle, the muscles on this side of the worm have to contract, while the muscles on that side of the worm have to not contract. They have to relax. And that's a relationship. The muscles here are contracting, while the events here of muscular contraction, they're not contracting. So something has to relate the signals going to these two muscles. They have to be linked, and they have to have this relationship. And this is one of probably the most basic relationship in muscular coordination, but in fact it's also used, it's similar relationships between muscular contraction allow you to have sensational performances such as this one. The point is, it requires coordination between events. That means the events must be connected somewhere in the brain, and the right relationship between those muscular relationships, muscular contractions established. So it's a connection and a relationship. This is sensory motor coordination, This is a picture from Mark Kanishi of a barn owl coming down to get a mouse. The mouse is shown here. This is taken in complete darkness using infrared. It was done in the 1970s. And it actually repeats an experiment done by Roger Payne in the late 1960s. And you'll notice that the mouse is actually going to escape the clutches of the barn owl, because the barn owl is going to go and strike at this little thing. And this little thing is a piece of paper that's tied to the mouse's tail. And the mouse is walking on sawdust, so its feet make no sound. But as the paper is dragged along, it rustles. And the owl hears the rustle, doesn't hear the mouse, so it strikes at the rustle. But obviously the mouse has to detect an auditory pattern. That must connect events. It must connect this auditory pattern with its own body coordinates, and it must then coordinate its muscles and in order to intercept the piece of paper. And that's connections and relationships. And here in sport, you can see the same thing. And here you can see a hawk moth inserting its proboscis. You probably can't see a tiny thin tube into this flower to extract nectar. And the common thing about all of these is that they require object identification, pattern recognition, spatial location, prediction, knowledge of body position, control of body posture, and the coordination of muscles. And that is all to do with linking together events, either sensory events coming in to determine their patterns or motor events going out. So we just think about pattern recognition. There's Necker's cube. You see that as a cube. Your brain is actually actively constructing that pattern because it's a bistable perception. Here is selection of cheeses, which you recognize as a relationship between a set of events, the different olfactory receptors which are being stimulated by these different cheeses, and the different gustatory receptors when you come to taste them. Here is a familiar thing that if you've ever seen this, you immediately recognize it, but when you first saw it, you didn't. And that shows that you've been able to connect these dots together to make a pattern, to assemble a pattern, and you've actually remembered it. And finally, this is a piece of music where you recognize from the visual pattern that there's going to be an auditory pattern. Again, these are sequences of events put together and coordinated in a particular way. And you can then transform that into a piece of music if you're able, in this case, to play the bassoon. Communication supposedly depends on people connecting and relating to each other, but it's obvious that in order to communicate, you have to produce a signal, and that requires that the components of the signals, which are events, are connected together in the right way, in time. And it also has to be interpreted by the receiver, which means that they must detect this pattern by connecting together the events to show that it corresponds to the signal. And you can see this, it's found in birds. There's a cricket, electric fish communicating with electric signals. Those waveforms are spectrograms, so they're singing to each other. This is a peacock making a visual signal, and this is my favorite, which is an Australian jumping spider, which not only has a peacock's tail, the male has a peacock's tail that he raises up to show the female, but you can see he also waves his arms around to make little semaphore, and there's a lot of connections of events going on in that. And learning is connecting. One event to another event, the present to the future, and the past to the present. And learning, this is Pavlov's dog, where Pavlov is looking at the dog, which learns to associate the ringing of a buzzer with the presence of food or the delivery of food. So it salivates, ends up salivating when the buzzer's rung, something it didn't previously do before because it only salivated to the food. And this is Carl von Frisch training bees to go to nectar, which has been labeled by being placed on a card of a particular color, and it's surrounded by cards of all different forms of gray. And he reasoned in 1914 that if bees had no color vision, they would see this blue as a shade of gray, like in a black and white photograph. So if you put enough gray shades around them, sooner or later some of them would go to the gray and not to the blue, and they all went to the blue. And so this is associative conditioning. And if you look at the neurons, you can see that their branching patterns show you that they've evolved to make connections. These are neurons that I've worked on, which are motion-sensitive neurons, and they're picking up local inputs from motion detectors to assemble a pattern of motion that corresponds to the optic flow generated by movement through the world. And so you can see that neurons are incredibly good at specializing to make particular patterns of connection. And of course, these are the connections that are the framework of behavior. But how do they establish the relationships? Well, very simple integrate and fire model neuron enables you to easily demonstrate that neurons are very good at establishing the sorts of relationships that are very useful for guiding behavior. So in this model neuron, the neuron receives inputs from spikes from other neurons, and they come to synapses and they produce these synaptic potentials here in the dendritic tree. And you have excitatory synapses, which produce positive changes in membrane potential. And you have inhibitory synapses, which produce negative changes, which oppose them. And the balance of excitation and inhibition determines whether the neuron will produce an output, which is an action potential. If the excitation is sufficient, then it will fire one of these action potentials. And if it's maintained or it's very strong, it'll fire several. So the neuron is taking inputs, connecting them together, integrating them, and then sending out a signal which depends on the relationship between these inputs and the threshold at which it fires. And then it distributes this, it makes more connections, it distributes this to other neurons. And in fact, this very simple model, and I've warned you beforehand not to believe that the brain was full of simple model neurons, but this is a very basic process. This simple model is not so far from reality. This is an experiment from Carl Peterson, and it shows what happens if you're recording from a pyramidal neuron, probably in the barrel cortex, the whisker cortex of a mouse, when you waggle the whiskers backwards and forwards. And on this rather complicated trace, the green trace is the position of the wiggle of the whiskers, the angle they subtend. And you can see they're being wiggled backwards and forwards. And this black trace is a recording of the summed signal from all of the synapses coming into the neuron. And you can see at this point here, the neuron fired one of these action potentials. And that's because it exceeded the threshold for generation of action potentials. So this sort of really does go on in the brain. So very quickly, let's just see what you can do with this type of neuron. So let's connect A with B. And now we make A and B produce action potentials, synaptic potentials, which are individually below threshold. But if A and B occur together, it will fire an action potential. So the relationship is that A and B coincide, and that's used a lot. You can use coincidence to learn relationships, to establish coincidences and learn them. And this is associative learning. And the way in which this works is that you have a neuron which responds to the meat, the food, and the food produces a large positive going set of synaptic potentials and generate a nice robust action potential, kind of what you would expect. Whereas if you buzz the buzzer, you only get a very small little potential, not enough to generate an action potential. Now, an action potential is a big event. It's a large signal in the neuron. And that large signal can cause internal changes in the neuron, which means that the action potential sends a signal back to the synapses, which is internal in their cytoplasm. And that signal primes the synapses, and what it says is, if in the previous few milliseconds, or 50 or 100 milliseconds, you received one of these little excitatory inputs, then the next time you receive an excitatory input, you should make it bigger. And that involves having a little memory of whether you received an input or not, and then saying, I've made an action potential. If you've got this little memory trace here, then reward the synapse, make it grow a bit stronger. And in my next lecture, I will show you how this sort of thing can in principle work. So now let's see what happens. When you first of all put on the buzzer, you get a weak signal. You follow it with the food. Now that's going to prime the synapse to increase. It's going to make the synapse increase so that the next time you stimulate, you get a bigger PSP. Pair again, bigger again, pair again, bigger again. And finally, you get such a large PSP here that the buzzer alone produces the spike. And this is an example of what's known as Hebbian synaptic plasticity, which was suggested from first principles by Donald Hebb in the late 1940s and was discovered actually working in hippocampus by Lomo and Bliss in 1973. So it's really something that's there. And it depends upon the neuron having an internal program of its own. It's not just a passive receiver and transmitter of signals. It can change the way that it receives and transmits. So let's go on. You can see that you can have, this connects A and B, but they're now, one is inhibitory and the other is excitatory. So to fire one of these action potentials, the excitation at A must be quite a bit stronger than the excitation at B. So the relationship here is that input A is stronger than input B. And that's a very useful thing to be able to say about events around you to drive your behavior. And in fact, this sort of neuron is used to inform the brain as to whether the sound from your right ear is louder or dimmer or softer than the sound from your left ear, so that you can localize the source of sound in space. And finally, you can use it to make decisions. Here, you've connected the neuron to all of the inputs which might favor you doing something. So this is the evidence coming in for doing X, and these are all the factors that would contribute to strengthening the evidence to do X. And just by simply activating more of these inputs, the EPSP gets bigger and bigger till eventually there's enough evidence and the neuron fires. And if you put in some inhibitory synapses as well, you can apply negative weights. Well, actually, I really don't want to do this. I don't want to lift this leg off the ground if the other leg's already off the ground or something like that. Okay, so that's decision making. And finally, the motor coordination that we looked at, where the worms in space, there are motor neurons which cause the extensor muscle to contract, extensor motor neuron, and a flexor motor neuron causes the flexor muscles to contract. And what you want to happen is you want to forbid the co-contraction of these two muscles. And so you have a simple circuit in which the motor neuron, when it's excited, sends a spike to contract the extensor muscle. It's also synapses onto a neuron here and causes that neuron to fire an action potential. But that neuron is an inhibitory neuron which prevents this neuron from firing an action potential. So you can no longer have co-contraction. And of course, you want to be able to do the same thing when you go ahead and now flex. And so you make a mirror image circuit, and these inhibitory inputs cross over. And so this is known in the trade as cross inhibition. And this circuit motif is found in virtually every animal's motor system at the level of the motor neuron. And so again, we have a connection between the motor neurons of the extensor and the flexor, a relationship that they're imposed on them, that both motor neurons are prevented from contracting both of the sets of muscles at once, and the relationship is being enforced by a pair of inhibitory neurons, which is now, you can see, is a simple circuit. but it's purposeful. And purposeful circuits will be the subject of my final lecture. So the conclusions are that neurons are actually using the energy to make the connections and establish the relationships that are behavior. And that much of the energy is consumed by sending signals along the wires that make the connections, and some of it to process at synapses the signals to establish the relationships and then take action on them. This purposeful processing is achieved by wiring up neurons in purposeful circuits. And processing is dynamic. I've given you just little snapshots of things going on in time, but really these are continuous processes. They're much more complicated. Processing is dynamic, and circuits are complicated because behavior is dynamic, and behavior is complicated. And I would say at this point, as a cellular neurobiologist interested in all of this stuff here, I feel we don't actually know enough about behavior. Cellular neurobiologists and biophysicists, people working on neural circuits, we're assembling really detailed, sophisticated accounts of interactions between neurons and what we do, and we do not have the equivalent in-depth understanding of many of the components of behavior that we're trying to explain. But there are changes on the way, and I think people recognize this by trying, for example, to use machine learning techniques to get unbiased measurements of behavioral relationships, which is what we need to know. But the most astonishing thing, I think, is that to generate behavior, neurons have evolved to be the most highly dynamic and connected cells in the body. They really are extraordinary cells. And a final thing from our energy budget. When David and I added up all of these quantities of energy, these quantities of energy, the spikes and the synapses, all depend on the generation of an action potential. And if you double the rate at which these neurons are going to be producing action potentials, you double their spike consumption and you double the rate at which their synapses are activated. And from that, knowing the total consumption of energy in cortical grey matter, you can work out, on average, in cortical networks, how often can neurons fire, averaged across all neurons. So this is a very broad global average. And the answer came out that it was less than 5 signals per second. So that doesn't say that there are neurons, that all neurons must stay below 5 signals per second. For short time intervals, they can produce many, many more spikes. But if one neuron produces lots of activity, another neuron somewhere else must shut up to keep within this limit. And what this means is that the signal traffic in the brain, that the rate at which we can process things and the number of events that we can connect and the relationships that we can establish are limited by energy consumption. And so there's a powerful selective pressure, Darwinian selective pressure, to evolve efficient circuits and efficient neurons in the brain. So it pays to be an efficient brain. And by looking at efficiency, one can see some new general design principles about how brains and circuits work. And that will be the topic of my next lecture. Thank you.
Right, the hour's late, but maybe just 2 questions? Yeah.
At the beginning, you mentioned that we're limited in being able to see activity of individual neurons with the bulb signal. Can you conceive of anything that might give us firing of individual neurons?
So people can see. So people, before people were discovered bulb signals, people were routinely sticking wires in and electrodes in and getting signals from single neurons. The real problem is that you want to know how neurons are talking to each other, and then you need to see the signals of 2 neurons that are actually connected. And in networks where the circuits are all interleaved, you have to record from large numbers of neurons to find out ones that are working together. And at the moment, people can record from getting well over 100 neurons at the same time, and relate their activity, but they're working in volumes of brain in which they're of the order of thousands of neurons, and so you're missing something. And so we badly need new techniques which give you a more comprehensive coverage of neural traffic. And that's a really demanding technical issue. Yeah.
So I didn't quite follow. So the 5 spikes per second per neuron is an average.
That's an average over space and time.
Exactly.
Yeah.
So, and I didn't know how you got from there to inferences about maximum firing, because as you say, there'd be selective trade-offs for extremely high rates if there's some emergency or,
yes,
and I didn't know if you thought that too much, whether there's cytotoxicity that can, come out from, anyway, just comment on where does the limit come from? Is it just the maximum amount of glucose and oxygen being delivered to the brain?
That's a very good question. So the single neurons have their own mitochondria and they generate their own energy. And if they fire extremely at high rates, they're going to need more mitochondria and be larger neurons to generate that energy, unless there's some way of sharing energy very quickly, ATP shared between neurons or something, and that's an open question. But some evidence, which I'll give you in the next lecture, suggests that indeed higher rate cells tend to be larger. And so the ultimate limit in that case would be the ability of the neuron to sustain the energy supply demanded by its high firing rate. But it'd be nice to know much more about the cellular physiology of energy demand and supply within single cells, and that's a difficult problem. That's a very good question.
All right, I think at this point we'll thank Professor Laughlin for his seminar.